So good evening, everyone. It's evening here in the United Kingdom, and welcome to our webinar on Ketosis and Crystals. Can diets help treat polycystic kidney disease? I'm Tess from the PKD Charity in the UK, and I'm delighted that Thomas Weimbs from the University of California, Santa Barbara is with us here today. His recent research and publications have sparked a huge amount of interest around the world, and we have nearly 300 people registered for this webinar. Before I hand over, I'd just like to run through some housekeeping, and then we'll start. So first of all, for those of you who've not used Zoom before and want to know how to ask questions, have a look for the Q&A box, which should appear somewhere on your screen. Type your question in there. You can type it in anonymously, and Thomas and I will see the questions, and Thomas will answer them as they start to show up on the screen at the end of his presentation. Now, because there are so many registered and we're expecting lots of questions, we may not be able to answer them all tonight. But if you do have additional questions, please do send an email to me at info@pkdcharity.org.uk. We'll do our best to get them answered. We will be recording the webinar and upload it to the site, the PKD website, for later viewing. Before I hand over, just to remind you of a few points and give you a medical disclaimer. So this webinar is for information only. It's not medical advice. We're making every effort to ensure that the information provided is correct and up to date. We do not accept liability for any errors or omissions. Advisors will not be promoting or recommending any treatment or brand. And always remember to consult your general practitioner, pharmacist, or other medical professional if you've got any concerns about your own care, your child's, or before taking any treatment. Just to remind you what our role is, we're here to support patients and families affected by PKD, raise awareness and fund research. Now, I'd like to hand over to Dr. Weimbs.
Great. Thank you, Tess. Welcome, everybody. Good evening in the UK, or good morning, good night, wherever you are, in which time zone. All right. So I just wanted to give everybody some links where you can look up more information. So this is the link of my lab website. Feel free to go there, look around. If you go to the publications tab on there, there you can access all of the publications from my lab for free. So you can download the PDF files. Feel free to go there and look at everything you like. All right, let's see. Another way of getting information is by going to my lab's Facebook group. So the address is up here, or you just, if you're a Facebook user, just type in Weimbs Laboratory and it should come right up. Feel free to join this group. It's a public group, and if you're a member of the group, you'll receive occasional news and updates, from new things coming out of my lab. So feel free to join there if you want to stay up to date. Good. All right. So quick announcement, as Tess already said, it's very important, before changing your diet or taking any supplements or medication, everyone should always consult their doctor or healthcare provider first. And that's, I think, very straightforward general advice. All right. So in today's presentation, I wanted to sort of introduce the problem of PKD therapy a little bit, and this is going to be the introduction up here. And then we have sort of two diet stories to tell, if you will. One of them deals with ketosis. So I'll talk about this first. The other one deals with what we call crystals, and I'll deal with that second. All right. So the first story is a diet story 1, ketosis. We recently published this paper on this. Again, you can find this paper. It's a paper in the scientific literature. You can find it on my lab website here. You can You're free to download the PDF file and read it. It's a paper that came out in printed form in December 3, so just a few weeks ago. It was already out online in October and started to generate a lot of kind of excitement and news in the field. Came out in a journal called Cell Metabolism, one of the top journals in the field of cell metabolism. All right. The paper made some news, and you can again get these sort of news articles from my lab website. They might be a little bit easier to digest if you're not a scientist and you don't want to read the actual original paper. Feel free to read those. So there was a news release by UCSB, a review in a journal called Nature Reviews Nephrology, another short summary in a journal called F1000 Prime. So feel free to take a look at those. For the second story, the crystal story, we also published a paper just a few months ago, October 2019, in a journal called the Journal of Clinical Investigation. Many co-authors on here. You might recognize a few names. Perhaps you have heard the name Boris, another leader in PKD research, Michael Mrak, another PKD researcher that's you might be familiar with, and many other experts are on this paper. Again, this paper also caused some news, and again, you're free to, welcome to look up these sort of summaries, which are more in lay terms and geared towards lay audiences. All right. So where am I? I'm at the campus of UCSB, University of California, Santa Barbara, and in particular in this building right up here. So that's where I'm sitting right now. I've been working on polycystic kidney disease for about 20 years. I started when I started as an assistant professor, starting my first lab at the Cleveland Clinic in Cleveland, Ohio, and then about 14 years ago or so, and moved on my lab and research over here. The current team members are shown here. The person that has done most of the work for these diet stories is my postdoc Jacob Torres. He's the first author on these two papers. And, we have, of course, additional grad students. We have many undergrads in the lab, and sort of education of the next generation of scientists is one important mission of the lab. The slide lists some of our recent collaborators, our current funding. And okay, let's dig right in. So PKD, I'm pretty sure most of the folks in the audience know a lot about polycystic kidney disease, so it keeps us really brief. You know that in the end there are cysts in PKD. I'll just close this here. And these cysts are what is causing many of the problems with regard to renal disease. And these cysts originate from what's called renal tubule epithelial cells. So as you may know, the kidney is made up out of thousands and thousands of these tubules. So they're really, as the name says, they're just like little tubes. They eventually connect all to each other and connect and empty out into the bladder. These thousands of tubes are in turn made up of cells, a single layer of cells. And in polycystic kidney disease, some of these cells start to proliferate, aberrantly. So they form these dilated tubules, and they keep on growing and growing to eventually form these cysts. So there can be thousands of cysts in each kidney. And these cysts keep on growing over years and decades. And in the process of growing, these cysts kind of compress and destroy the normal kidney tissue slowly. All right, so how do we study this? How do we study PKD? So one, that's just how most people do it that are studying diseases. You have to do animal experiments, and this shows an example of how this looks like. We actually started out, some of our first discoveries were that a certain pathway called the mTOR pathway, doesn't matter what it is, but we discovered that the mTOR pathway is strongly activated in polycystic kidney disease and in these cysts, and drives the cyst growth in the disease. And this became immediately very important because there was already a drug available called rapamycin, and there's similar mTOR inhibitors available. We tested rapamycin to see if it might inhibit cyst growth, and it did. So this is an example of one of our papers from back then, just to kind of highlight how these experiments look like. You see here on the left a cross-section of a normal mouse kidney, and in the middle you see a cross-section of a polycystic mouse kidney. This is from a genetically engineered mouse model, and you can see this cyst is much bigger than the control. This kidney is much bigger than the control kidney. You see all these cysts, which are essentially the white spaces here, and there's very little normal kidney tissue left in this highly cystic kidney. And if we when we treated these animals with these cystic kidneys, we treated them with rapamycin, their kidneys looked like this instead of that. So you can see there's a very big difference, and we can zoom in microscopically, and we can stain for certain things. In this case, blue is bad. Blue means fibrosis. So you can see there's less blue here, there are less cysts, and much more normal tissue, renal tissue left. And we can quantify things in any number of ways and look at molecular pathways. One convenient way of quantifying these effects is by just weighing the kidneys, and that's the 2 kidney weight as a % of total body weight ratio. That's what this means, 2K, divided by TBW, total body weight. And you can see these cystic animals have these very large kidneys, that take up about 6% or so of the total body weight. And if we treat these animals with rapamycin, it very strongly inhibits cystic progression in these kidneys. It also rescues their kidney function and looked fantastic. We published these data, and so you can see this just highlights how what the difference is between untreated and treated animals. And these results quickly led to clinical trials to test whether mTOR inhibitors may be effective in PKD. And some of you may remember this or may know about these trials, and unfortunately they came back disappointing. The 2 largest trials were published in 2 back-to-back papers in the New England Journal of Medicine. One was done in Switzerland, one in Germany. And essentially the result was that the mTOR inhibitors did not really lead to any clear beneficial effect. And in hindsight, the reason was likely that in humans, you just cannot use high enough doses of these mTOR inhibitors to actually inhibit mTOR sufficiently in these cystic kidneys. And the problem is that these mTOR inhibitors are very strong drugs. They're strong immunosuppressive drugs. So they have all kinds of extra renal effects that you don't actually want. And simply increasing the dose would not be a solution, because this would make patients just dropping out, would make them drop out of the study due to all these side effects. So this seemed to be the end of the road for mTOR inhibitors, unfortunately. But it also made us think, and others think about what are the challenges for drug therapy in PKD. And so we recently published a paper where we kind of discuss some of these challenges and some of the drugs that are on the horizon. So one problem in PKD in particular is that therapy would likely have to occur over a very long period of time, and we're talking years to decades. And it's just because it's such a slowly progressive chronic disease. Moreover, therapy would need to start early on, so that there's a chance for any therapy to actually save good kidney tissue and have sort of an effect in delaying the progression of the disease, which means that very likely one would have to treat relatively healthy people even long before they have significant symptoms. Which means there's a bit of a practical and maybe ethical dilemma, because you don't really want to treat someone who is healthy with a drug that makes them less healthy. Which means that probably any drug that would be feasible in the end would have to have an extremely low extrarenal side effect profile in order to make this function. All right. Another complication is that most of the potential what's called drug targets that have been identified by my lab and many other labs around the world, most of these drug targets are not really specific to polycystic kidneys, but those are proteins and signaling pathways that are just needed everywhere in the body in many other tissues and organs, which means that if you inhibit them with a drug, very likely do you not only have beneficial effects on the kidney, but you also would have potentially detrimental effects on many other organs and tissues. So we wondered if drug side effects are the problem, is PKD therapy possible without drugs? That would be, of course, fantastic if that were possible. And so this leads into these two diet stories. So let's start with the ketosis story first. And this story really in my lab started with a former grad student of mine who has since graduated. He got his PhD. He started a new job at a biotech company. And his name is Kevin Kipp. So he, while he was in my lab, came up with this idea of feeding our PKD mice less food. And he had some mechanistic reasons for it. He thought that this could lead to mTOR inhibition. And I would never have thought that this works. And luckily, he didn't actually ask me. He just did it and came back with the results. And it actually worked, to my great surprise. So I'm glad he didn't ask me, and he went ahead and did it anyway. So what he did is he essentially had a experimental cohort of these PKD mice and gave them 23% less food on average, compared the cystic progression to an ad libitum fat control group, and found it actually worked. And the way these feeding experiments work is you weigh, you measure how much food an animal eats per week. If you give it to them ad libitum means just free access to food. So they have a mountain of food in their cage. They can eat whenever they want, however much they want. We just weigh out, how much they actually ate per week. And that's the curve up here. So that's what the control group consumed in food. And then my student just gave the experimental food about 23% less. And that's how all these types of caloric restriction studies are done. And you can see with this very mild reduction in food intake, there's actually no change in body weight gain in these animals, so they're not malnourished or anything like this. Yet, surprisingly, there was a very strong inhibition of cyst growth and cystic progression in these animals. So here on the left is a normal mouse kidney, in the middle is a polycystic mouse kidney, and on the right is a polycystic mouse kidney from the cohort that was fed less food. And you can look at this microscopically. You can weigh the kidneys again and express it as a weight per body weight. So the ad libitum fed animals had these very large kidneys, and this was brought down very significantly in the reduced food intake, or RFI group. It also led to sort of a partial rescue of their kidney function, and everything we looked at looked much improved. So we published this paper essentially saying that a mild reduction in food intake slows PKD progression in this mouse model of PKD. And we essentially scratched our head and couldn't really explain why it worked. But we published the result, and out there it is for everyone to see. And at the same time, very interestingly, another group completely independent from ours, and that's a group at the Mayo Clinic, published a very similar paper. So they had a very similar idea. They reduced food intake in their PKD mouse models, and they started out with a 40% reduction, saw a great beneficial effect, and they titrated it down to 10% food reduction, and they still saw that it works. And they published a paper again saying it works, and we don't know why. So neither of us really could explain why it was so effective. And so this was a few years back, I believe in 2016. Since then, we tried to answer the question, why is this so effective? So is it due to caloric restriction? Does it have anything to do with the reduction of a certain nutrient group, maybe carbs, maybe proteins, fat, or anything like this? Or is anything else going on? And so we have since figured this out, we think, and published this paper very recently. And this was done mostly by Jacob Torres, with help from others in the lab, including Sammy Kruger. And the explanation is maybe best illustrated in a cartoon like this. So what happens in the control group of animals, and actually the explanation has to do with sort of a behavioral change in mice, in the way that these feeding studies are done. So the ad libitum fed mouse population have free access to food 24/7. So they essentially are like a Jerry mouse living in a cage of cheese, and they will do exactly what you think they would do. They nibble all day long, they eat a little bit here and there, and eat all day long. In contrast, the animals in the reduced food intake cohort, they only get their food once a day. It's put into the cage and it's pre-weighed a certain amount of food. They get it once a day. And now these mice behave very different. They're thinking, I'm assuming, they're thinking, hey, if I don't eat this right now, the cat is going to come and chase me away and my food is gone. So what they do instinctively is they eat all the food within 1 hour. So we put breakfast, lunch, and dinner into their cage, and they eat everything in 1 hour, which means they're, temporarily get very fat. They're actually convert most of the carbohydrates that they ingest into fat, into their fat deposits. And then for the remaining 23 hours, they're actually not eating anything and they're starving because they ate it all already. And what happens during this starvation period is that the body really changes its metabolism and now digs into the fat reserves. And that's a process called ketosis. So what is ketosis? I'm sure most of you have probably heard about it. And here's sort of a physiology 101 recap. As you can tell, I'm a biology professor. So what is ketosis? So during our normal, feeding, if we're eating lots and lots of food day in, day out, the molecule in our blood that provides most of the energy for our organs and tissues and cells, that is glucose, also called blood sugar. So all these tissues in our body, they just take up blood sugar, they burn it, they use it for their energy needs. And any excess of blood glucose, anything we don't utilize, is converted to fat in our body fat or adipose tissue, and it's just stored there. And that's what just normally happens when we eat. What happens when we fast? So during fasting, our the blood glucose supply goes away, and our glucose, our carbohydrate stores in the body can be quickly used up, within 24 hours or so. So you would think our organs and tissues and cells might be in trouble because now there's no more energy. But of course, they're not in trouble because evolution has come up with this whole idea of the fat stores. So during fasting, the fat stores release the fat in the form of what's called fatty acids. They're released into circulation, and the cells in our body can use the fatty acids as an energy supply. And some of the fatty acids are also converted by the liver into another class of molecules called ketones. And these ketones can also be taken up by all these cells and tissue for energy. So during fasting, our body essentially switches from utilizing glucose to utilizing fatty acids and ketones instead. And this process is called ketosis. And it's a normal physiological process that all of us should undergo, pretty much all the time, because that's how evolution designed us to be. Interestingly, ketosis is something that is often mixed up with a very similar sounding condition that's called ketoacidosis, which is a pathological condition that can be very dangerous. It's not really related to the physiological condition of ketosis. So ketosis is something good that we can all do. We can go in and out of ketosis, and we should do that. Ketoacidosis is something completely different, but unfortunately, doctors often, if you talk to them about ketosis, they instinctively think, "Oh, ketoacidosis, dangerous, don't do it." So that's just something to be aware of. All right. So what about these ketones? One of the ketones, maybe the most important one, is called beta hydroxybutyrate, or BHB. So just put a pin in it and remember that BHB is one of these ketones, and I'll get back to it. All right. So what happens now during time-restricted feeding? So, which is essentially what we did in these mouse feeding studies, right? So we feed them every 24 hours. So we feed them here, we feed them there, and you can actually take these mice and measure their body, their total body metabolism. Never mind how this works or what the values here mean. It just means if the values are up here, that the mouse is burning carbohydrates. If the value is down here, the mouse is burning fat. And you can see every time you feed a mouse, these values shoot up and they start burning all these carbohydrates away until they're used up, and then they crash back down and start burning the fat. So for about half a day, they're burning carbs, the other half they're burning fat, and then you feed them again, and the whole cycle repeats. So this is just a very normal physiological process of going in and out of ketosis. So to test whether ketosis is important in explaining these beneficial effects that we had seen, we tested several different animal models of PKD, in particular a rat model, a mouse model, and a cat model. So all of these particular animal strains, they have polycystic kidneys, and the mouse and the cat model are what's called autologous models. So that means they have mutations or defects in the same gene that is affected in the human disease. All right. So I'm just showing a few bits and pieces of some of the experiments just to give everybody a flavor for how this looks like. So here we're looking at a rat experiment where we subjected the rats to time-restricted feeding. So they have access to food only for an 8-hour window in their 24-hour cycle here. And they have access to food in the dark because rats are night active. So that's when they would normally tend to eat. And the ad libitum control group has access to food 24 hours a day. And if you look, and if you do this for 5 weeks in these rats, and you measure the kidney weight as a % of total body weight ratio, you can see the cystic animals that are in the regular ad libitum feeding schedule, they have big kidneys, and the animals on the time-restricted feeding schedule, they have significantly smaller kidneys. They also have fewer cysts. We can look at all kinds of different molecular outcomes, and we can look at microscopy, and literally everything we looked at was greatly improved. Okay, so that suggested, let me just go back, that suggested that time-restricted feeding without any caloric restriction already has a beneficial effect. And I should mention that in this particular study, both groups, the ad libitum and the time-restricted group, they ate the same amount of calories. You know, at the end of the day, if you add it together, they eat the same amount of food, same kind of food, same calories. The only difference is really the time restriction here. All right, so we asked how else can we induce ketosis? And one other way of doing it is called the ketogenic diet, which is a very low carbohydrate diet, and it's very high in fat and moderate in protein. So it has sort of the normal protein content as normal red food has. And the only difference is really it's very low in carb and very high in fat. And if you feed animals a ketogenic diet, which is the orange line down here, you can see that you're essentially forcing them to burn fat all the time. So they never go out of ketosis. They're always in ketosis, and it doesn't actually harm them in any way. What we found near the outcome here was, again, a very strong inhibition of cystic progression in these rats. So here are again the ad libitum control rats, and these are the rats on the ketogenic diet. You can see their kidneys are much smaller, they're much less cystic. And again, we looked at fibrosis and you know, everything we typically look at, everything is improved. We repeated this experiment now here in this figure with older animals. So in the previous figure, these were juvenile animals. Now we're looking at sort of young adult rats that already have established cystic disease at the beginning of this study. So we start treating them at week 8 until week 12. So it's the week after birth. And at week 8, they already have cystic disease. You know, their kidneys are about 40% cystic. And if we keep them on a normal diet, that doesn't change very much. But if we put them on a ketogenic diet, you can see the proportion of cystic tissue actually decreases, suggesting that there's a regression of existing cystic disease. And you can also see the total kidney weight goes down after being on a ketogenic diet for this period of time. So this was actually very surprising, because usually that's what we would not expect to see with any drug treatment we and others have done. You typically don't actually see a regression of the disease. You know, the best you can hope for is sort of a stop of or halting of the further progression. All right. Another way of a dietary intervention that leads to ketosis is just acute fasting. So just like straight out fasting. So we give the animals no food whatsoever. They still have free access to water. So they always have access to water, but here in this case, we are using cats with polycystic kidney disease, and we fast them for 72 hours. And this was done by our collaborator, Leslie Lyons at the University of Missouri, who has these cats, and she identified the mutation in these cats. Turns out that cats of Persian cat origin have a high incidence of polycystic kidney disease, and it's actually due to a mutation in the same gene, the same PKD1 gene that's affected in most human cases of PKD. And this is actually the first interventional study in PKD cats out there. So we're very happy to collaborate with Dr. Lyons on this. So this is a study using 4 of these polycystic cats, and we measured their total kidney volumes by CT scanning. Some of you might be familiar with how these CT scans look like. Obviously this is a cat now, not a human, but you can see here's the torso of a cat. There actually the 2 kidneys. There's one, there's another one, and you can see there's some very large cysts you can easily identify here. And we can measure the total kidney volume off of these CT scans. And this particular cat, if we measure the total kidney volume about 17 months before the fasting study, and then just the day before the fasting study, you can see that during these 17 months, this particular cat had this much of a progression of their cystic disease. And then when we fast them for 72 hours only, there was a very dramatic a decrease in total kidney volume. We saw decreases in all of these treated cats, in these fasted cats. Some showed a larger effect, some a lesser effect. And the rapid decrease appears to be due to essentially as a draining of individual large cysts during this very rapid time period. So this was a kind of a dramatic result, which again, we didn't really expect it to be that dramatic. And we got essentially the same result in our rat and mouse models as well. So very rapid regression of their cystic burden. Okay, so we talked about what ketosis is, and that during this fasting or intermittent fasting, or if you ingest a ketogenic diet, you essentially have high levels of fatty acids and ketones. And we wondered if perhaps the ketone, especially the ketone called BHB or beta hydroxybutyrate, if that might actually be responsible for at least some of these beneficial effects. So we tested this directly and fed our, in this case, the rat model again. We gave these rats BHB in the drinking water. So we take this ketone, which is a naturally occurring ketone that comes up during ketosis, and we put it into the drinking water and treated them for 5 weeks. And got a very dramatic result. So here is a typical cystic kidney from this rat model. Here's a control kidney from a non-cystic rat. So you can see the big difference here. And this is a kidney from a cystic rat that was treated with beta hydroxybutyrate. And remember, a postdoc Jacob Torres, when he got the very first result back, he came to my office in a hurry and said, "Hey, Thomas, this looks really interesting. I don't know if I believe it yet, but these treated cystic kidneys, I cannot tell them apart from the control kidneys." So he had to go back and double check and triple check everything, and it was correct. So we see a very dramatic effect of treatment with BHB, at least in this rat model. And you can see, when we quantify this again, so that's the kidney weight as a percent of total body weight of the control cystic rats. And if we treat them with BHB, the kidneys are much smaller. And again, we can look at fibrosis and all kinds of parameters, and they all appear to be greatly improved. All right, so what are the conclusions from this? We conclude that cyst lining cells, so the cells that actually make up these cysts in PKD, that they are metabolically inflexible, that they are highly dependent on glucose, and they cannot deal with this metabolic switch that occurs during ketosis, suggesting that they cannot metabolize fatty acids and ketones, and they just need the glucose. Another conclusion is that if we actually subject PKD animals to ketosis, we see of course, a reduction in the level of glucose, suggesting that these cystic cells would have less energy to proliferate. But also what occurs is this increase in fatty acid supply. And we have some indications that these cystic cells, they can load themselves up with fatty acids, but they cannot metabolize them. So they essentially poison themselves with too much fatty acids. And furthermore, the ketone beta hydroxybutyrate appears to be very important, and as an important mediator of this beneficial effect, either due to a metabolic or signaling effect, and that's something we're working on right now to nail down the exact molecular effect. All right, so we published this paper and it made a big splash because the implication is that potentially ketosis or any dietary intervention that leads to ketosis may be, potentially be used as a therapy for PKD. Just to discuss this for a moment, so fasting can probably be regarded as the oldest form of medical therapy, dating back to prehistoric times. And, we all know that fasting plays an important role in essentially all of the world's religions for all kinds of reasons, and is kind of the most prescribed therapy, in the good old days, in medieval days when doctors had nothing else to do to give their patients, and the first thing they usually prescribed was fasting. This has gone out of fashion, and these days doctors don't prescribe fasting very much anymore at all. But it's important to know that this is actually a time-honored therapy, if you will. And then dietary interventions such as time-restricted eating, and you may have heard of a 16-8 diet or alternate day fasting and so on. They're actually fairly widely used by many people to control diabetes and obesity. There are ketogenic diets that have actually been used to treat childhood epilepsy for about 100 years already, very, very successfully. In general, these dietary interventions are, very widely, of course, available. You can always eat less. They're very inexpensive and safe for most people, at least for healthy people. So there's something to be said for, people with compromised kidney function. There are many different considerations in those populations. All right. However, as we all know, it is very difficult for people to adhere to diets. So if it were easy to adhere to diets, there would essentially be no diabetes epidemic out there, which is probably entirely curable by just dietary restriction. So that's why we're actually very excited about the finding that just supplementation with the ketone beta-hydroxybutyrate appears to be able to mimic most of the beneficial effects that we can see. All right, so, and this sort of a picture that one of my very talented students drew, how she imagined how our animal fasting study went, and you can see all these disappointed animals when the diet of the day is revealed to them, which is nothing on their plate. But very important, they all have free access to water, and that's something we always want to make sure of. All right, okay, very quickly, diet story 2, so much shorter version of this here, and a summary of it. Okay. So again, we published this paper just a few months ago, and essentially the bottom line of this paper is shown in this cartoon here. So we all learned already, and we all know that the kidneys are made up of about 1 million or so of these tubules, and there's kind of fluid flow going right through the center of these tubes, which eventually all collects together, and that's what becomes the urine in the end. And you may know that the kidney is always challenged by these insoluble compounds that tend to be excreted through the urine and through the kidneys, but they also tend to be very insoluble, and they like to precipitate and form crystals. And one class of, or one molecule is called calcium oxalate that comes from oxalate, and a lot of the oxalate originally comes from our food, from our diet. And another source is calcium phosphate. And again, phosphate can be controlled by diet. There are certain foods that are very rich in phosphate, which means the kidney has to excrete a lot of the phosphate, and that often tends to just precipitate and form these crystals. Another compound is called uric acid. Again, it's diet controlled. If we eat a lot of meat, or if we have gout, there is a very high uric acid level in our body that all needs to be excreted through the kidneys. And the challenge is, that the uric acid again wants to form these little crystals. So these crystals form in kidneys of just normal people all the time, and all of us are excreting millions of these crystals every day with the urine, and it's just a normal process and usually causes no harm. And every once in a while, some crystals can get stuck in these tubes. And maybe it's because we ate too much oxalate in our diet, and there's just too much coming through. The kidney can't handle it at this moment. Crystals get stuck, they get lodged in these tubules. And it had been unknown how these lodged crystals really get out of there after some time. So what we discovered, and that's described in this paper we published, what we discovered is that these lodged crystals cause activation of certain signaling pathways, which leads to the dilation of these tubules. So they just widen the diameter, which then lets these tubules to flush out these crystals. And after about a week or so, everything goes back to normal. So these kidney tubules go back to their normal diameter. And, if you're the owner of this kidney, you would never know anything about it. So that's just a normal process for how the kidney can deal with these crystals. What happens, however, in PKD is that these dilated cysts do not appear to be able to go back to their normal diameter, and instead overshoot and form these cysts. Which means that the more crystals you challenge your kidneys with, the more cysts you get in the end. And we could actually show this in this case in a couple of different rat models. So here we're giving rats a precursor of oxalate, which is called ethylene glycol. So it gets metabolized to oxalate, has to be excreted through the kidneys. And you can see, the normal rat kidney, the cystic rat kidneys are here. If we give these rats ethylene glycol in the drinking water, their cystic progression is worse. So they get worse disease. And we got a very similar result, and that was actually done by Vicente Torres, our collaborator at the Mayo Clinic. They fed a different rat model a high phosphate diet compared to a controlled phosphate diet, and you can see the high phosphate diet made it much worse in these animals, and also led to calcium phosphate precipitation. All right. Look at my timing here. All right. Okay, let's speed up. All right. So the question is, does prevention of crystals in the kidney, does it lead to inhibition of PKD progression? And amazingly, these data have been out there already about 20 years ago. And it was probably not really realized at the time, or certainly not really realized at the time that it had anything to do with this mechanism I just told you about. So back in the day, about 20 years ago, a scientist by the name of George Tanner published a series of terrific papers, very solid data. And what they did is they treated PKD rats with citrate. So citrate or citric acid, just a normal a dietary supplement and a normal body product, turned out to be very effective, and it almost doubled the lifespan of these rats. It improved their kidney function and looked to be extremely effective. Unfortunately, they couldn't really figure out the mechanism why citrate was effective. And now in hindsight, we think the reason why citrate was effective is actually because citrate can inhibit these crystals from forming. So citrate just prevents calcium oxalate and phosphate crystals from forming in those kidneys, meaning that it can protect these kidneys and prevent the rapid progression of the disease. So unfortunately, these studies were published and then quickly forgotten. When things are published in the scientific literature, some scientists pick them up and look at them. And if it deals with anything such as citrate that you can't patent and you cannot do anything with. You can't really start a company off of it. There was no clinical trial ever done to test anything in humans. So this was all quickly forgotten, unfortunately. So I'm convinced we could have already potentially had a fairly effective treatment out there for maybe 20 years, and it hasn't happened yet. All right, okay. So practically, this might mean that potentially PKD progression can be reduced again by dietary interventions, for example, by limiting foods that are rich in oxalate, phosphate, and purines. So purines are these precursors of uric acid, and they occur mostly in meat, or is like more concentrated in meat. Another measure is to increase water intake, which just leads to the dilution of all these waste products in the kidneys as they pass through, and that gives them less of a chance to form these crystals. And I know I'm not giving any medical advice, but I think this counts not so much as medical advice and more as a no-brainer advice, perhaps. In knowing the study, I think it should be a no-brainer to cut out at least some of the very extreme oxalate-rich foods out of everybody's diet, such as spinach, which is kind of right at the top of the list. And there are lists you can find on the internet on which foods are very rich in oxalate, which ones are safe to eat, and so on. So I think unfortunately most nephrologists wouldn't necessarily know the latest publications yet. So chances are they would never have heard of this study yet. They may not be aware of it, but now you are. So maybe bring the study to your nephrologist next time you see them, and ask them, hey, should I really do this? Should I limit my spinach intake? All right. Okay. So that's it from sort of like just summarizing the scientific papers and studies we've published recently. And just to emphasize that all of this work was done in animals. And it's always a challenge next then to apply what we have learned in animals and apply that to humans and dietary recommendations for humans. But what you can do, and obviously we're working on many of these questions, including coming up with some really solid science-backed dietary recommendations. If you want to stay in touch and get news and updates from what's going on in my lab, feel free again to join my lab's Facebook group. Here's the team members. And let me get out of this screen sharing so I can see if there were any questions. Let's see. All right.
Thank you, Thomas. That was fantastic. So we do have some, can you see the Q&A? We do have a number of questions.
Okay, yes, I'm pulling it up.
You can answer them live.
Mm-hmm.
If you start at the top, and then if you just tick when you've answered them, you should be able to see the ones below.
All right, good. Okay, yes, so I'm happy to answer questions. Just as a disclaimer, I'm not a medical doctor. I cannot give any medical advice or dietary advice. And I don't want to promote any products, for example, but I'll try to answer questions that are, a little bit science related, perhaps. Can PKD patients really do ketosis diets safely? All right. So I'm not going to say yes, you can, no, you can't. However, I want to point out, after we published our paper, and it came out in October last year, very quickly, this was picked up by patient groups, and a couple of Facebook groups popped up very quickly where people started to share their experiences with ketogenic diets. We have actually also identified a whole number of patients that had already experimented with ketogenic diets, for all kinds of reasons, health reasons and so on. And many of them told us that they had really good experience with it. We're in the process of doing what's called a retrospective clinical study, where we are recruiting patients that have already experimented with ketogenic diets, for any number of reasons, and by questionnaire, just asking them what their experience was, positive, negative, was there any, detrimental side effects you've noticed? Was there any beneficial effects? How easy was it to adhere to the diet, and so on? That's a study that's going to be led by a nephrologist in my lab, and we'll put out a call to participate in the study very shortly. So we plan to publish this in the scientific literature as a case study, and I'm curious to see what we can learn from it, because I think there's, given the very large number of PKD patients worldwide, I think there's a fairly substantial level of experience already out there that we can maybe learn from. However, ketogenic diet does not equal ketogenic diet. So I think it's very important to not just go out there and pull up some ketogenic diet recipes off of the internet, because that may not, very likely, those would not have kidney patients in mind. Typically ketogenic diets are done for weight loss, for otherwise healthy people that have normal healthy kidneys, and there are lots and lots of different considerations to keep in mind when somebody has compromised kidney function, right? So then everything changes. The electrolyte handling is very different from somebody with healthy kidneys. So I would absolutely caution from just doing something completely on your own. That's probably not something you want to do. So that's what I'd say about it. And we're actually, we're very motivated in coming up with a sort of an approved ketogenic diet, if you will, or an approved diet in general. And we're talking to some experts, and are thinking of writing a book right now about all this, taking into account all the scientific findings that we have on the subject. All right. Okay. So, all right. One question is, do you know if it's safe to take BHB to increase ketones? Again, I cannot give any medical or dietary advice, but I want to absolutely caution here, because these ketones, you can buy them, and they're meant for athletes typically. Like a marathon runner might take those to boost their energy level. These are typically not necessarily safe for people that have to think about their kidney function. There can be things in these formulations that are not kidney safe. So I would absolutely caution from just doing this on your own. Just to let you know, we're actually in the process of starting a company, and I'm not promoting anything here, just for your information that there's something coming up, but we're coming up with a company to come up with kidney safe formulations of dietary supplements to promote kidney function or to support kidney function. So that's something on the horizon. All right. Is it suitable for PLD patients too? Is one of the questions. PLD refers to polycystic liver disease. Many PKD patients also have liver disease, on polycystic liver disease. That's one of the very common complications of the disease. We currently have no information on the effect of ketosis on cystic liver disease. Unfortunately, that's something we're very interested in. That's on our to-do list, and we're working on it. So I have no information. I would say stay tuned for that. It just so happens that the animal models we have been using don't have very prominent liver disease. And that's just how these models sometimes work out. So we're trying to answer this question about liver disease. So stay tuned on that. All right, let's see. So there are many similar questions. I'm just browsing through them. All right. Are there any cardiovascular issues for animals on a ketogenic diet? That's something interesting. People have the natural instinct of anyone that hears about a high-fat diet is, whoa, this will lead to cardiovascular issues. High fat equals bad for your heart and vascular tissues, and that's the typical instinct reaction of almost any doctor out there. If you walk into a doctor's office and say, hey, what do you think about a ketogenic diet? They will say, whoa, the high fat is just going to give you a heart attack. Turns out that's actually not the case. So there are some recent studies where essentially the opposite happens. So some of these ketogenic diets can actually be protective to cardiovascular issues. Mm-hmm. Okay, there's a question about the medical community and nephrologists maybe hesitant or reluctant to move into dietary approaches. Okay, yeah, and that's unfortunately, my colleagues, that are nephrologists, those are all fabulous people, and they have the best interests of their patients in mind. But you have to keep in mind, they're seeing patients with all kinds of kidney diseases. PKD is just one small fraction of them. They're typically wouldn't necessarily know, the forefront of, the scientific literature that's out there on every single kidney disease. So chances are your nephrologist may never have heard about these new studies yet. And that's very understandable. So I would say, that's an opportunity for patients to be an advocate for themselves and just, bring these studies to your nephrologist the next time you go and say, hey, take a look at this. Let me know what you think the next time I see you. And maybe, they might read it and they might come back with an answer for you. All right. Okay. Is it possible to correlate the stage of kidney disease in the rat or cat models? Good question. I would say it's very difficult to do that. You can't easily translate, the progression in these animal models to the progression in the human disease. There's no perfect model, no perfect animal model out there. There are many, many different mouse models, a whole bunch of rat models, and this one cat model. Typically, what we tend to do in my lab is we don't like to rely on only one animal model. So whatever we find in one animal model, we tend to double check that in an independent model. And only if we get sort of consistent answers between, two or more models, we tend to be, convinced that there's predictive value for the human disease. And that's exactly what we've done here. We've used a mouse, a rat, and a cat model. They're all led to consistent conclusions. So we think there's a reasonably high chance that whatever we found in these animal models may be predictive for human disease. But I wouldn't go as far as saying, this particular stage of the rat equals that particular stage of the human. You can't easily do that. All right, which also means, anyone out there that reads our animal study, I would caution against the temptation to say, hey, they did this in the rat, let me do the same for myself. So you should never do that based off on an animal study. All right, let's see. There's a question on intermittent fasting while being on tolvaptan. I'm assuming some, of the people in attendance may be on tolvaptan. Again, that's something I can't comment on. So that would be a question for your doctor. I should probably say that likely if one is in a clinical study, changing your diet sort of in a dramatic way might disqualify you from the study. So that's something to keep in mind. What are the challenges and chances of conducting human clinical trials regarding the impact of keto diet on Adpkd progression? That's one question, great question. Yes, so the challenges are diet is something inexpensive that the pharmaceutical company cannot make any profit of course. And as you probably know, most of the clinical trials are, funded by pharmaceutical and biotech companies, and for very good reasons. So they're coming up with new drugs. It costs them, on average about $2 billion, billion with a B, to develop a drug all the way into, all the way to the market. And part of the expenses are just that these clinical trials are so expensive, especially in a disease that's a slowly progressive chronic disease such as polycystic kidney disease. So we're talking a clinical trial typically takes something like 2 years treatment. So start to finish, recruitment and data analysis and staggered treatment of patients. You easily talk about something like 4 years for a clinical efficacy study. And they tend to be very expensive. Patients are usually monitored by MRI scan and many expensive blood tests and so on. So it takes millions and millions of dollars to actually conduct a clinical trial like this. We're actually planning a clinical trial with our colleagues in Germany. So it will be a much smaller trial, much shorter term trial, and essentially just a feasibility trial, which we hope to be able to, to get funding for, to be able to do this very quickly. It wouldn't be the end-all proof yet, because that would require a much larger study. We've been thinking of potentially getting a multi-center study like this funded by the different PKD foundations and PKD charities, let's say in Europe, to all chip in some funding for it, to be able to do this. So that's something that we're very actively trying to pull off. And that's typically the challenge, that these dietary intervention trials, they're very difficult to get funding for. All right. Of course, the chances of doing a trial like this would be that we would know once and for all, how effective is this. So that would be very important. The amount of citric acid required to prevent crystal formation. Can this be done through dietary modification, for example, eating lemons, limes, oranges, etc.? The question is, so again, I'm not giving any, dietary advice. I'm not saying go ahead and eat lemons, by the pound. And that's actually one of the problems. You might have to eat lots and lots of lemons or drink lemon juice to be able to do this. And, that's something that somebody might do for a week or two. And that's something that's very difficult to do, for the rest of one's life. So I would say, so that's kind of the point of this company we're about to launch, is to essentially come up with something that is much, much easier to take than eating, 5 pounds of lemon every day, just to pull a number out of my head. Okay, somebody's asking, during the fasting period, should we just drink water? Again, no dietary advice here, but just to emphasize, and I think that is just general advice for PKD patients, make sure you have a high water intake. And again, that also, of course, depends on the level of kidney function. So everything changes once kidneys stop functioning. But for anyone like well before that level, sort of an increased water intake, I think is very generally recommended by any nephrologist. And one of the reasons we think it is actually advantageous is because it will dilute all these crystal forming compounds and make it much less likely that these crystals actually form. So just for that reason alone, make sure to, when anybody is fasting doesn't mean no water. Fasting means no food, but water, you always want to keep that water intake. All right, okay. Let's see. So, Tess, what do you think? Shall we, it's already 18 minutes past the hour.
I'm just having a quick scroll down. Somebody did ask about what happened after ending the diet. Did the cysts then go back
in the
to
animals? Okay. Yeah, that's a good question. So what happens if we do like an acute fasting treatment in our animals and then put them back on normal food? Do the cysts quickly grow back as they had disappeared in the first instance? We're doing these experiments right now. I don't have the answer yet. So that's again one of these stay tuned answers, unfortunately. But that's definitely something we're very interested in knowing. So if we do repeatedly, if we do acute fasting sort of in a repeated fashion, where does this lead us? Do we have a sustained beneficial effect in this case? Good question.
Another question in the chat to do with the mTOR pathway, asking, is it involved in skeletal muscle growth and sustaining muscle?
The mTOR pathway in muscle. That's a question I don't know the answer to right now. So, well, mTOR is needed in many different tissues, not only the kidneys. Well, maybe the question is more related to fasting. I'm assuming that's where the person is getting at. That's one of the sort of misconceptions where people say, hey, if I'm fasting, doesn't it mean I'm losing my muscle mass? Usually it doesn't mean that, unless you're fasting for a really long period of time, like many days. So our bodies have really evolved, over, we are animals and we have evolved over millions and millions of years. Fasting is just a normal, it's just a normal occurrence for us animals. And, think about it as, back in caveman days, when we had to, hunt a deer and we had plenty to eat for a couple of days. And then it took another week until we can hunt the next deer. So we have a few days where we have to just fast. And that's just a very normal transition to go in and out of ketosis. That's what we should all be doing nowadays, but almost none of us is doing it because we have the convenience of our refrigerator and our pantry full of food. So most of us, start eating their breakfast. The first thing you wake up, you eat your breakfast, you have your lunch, you have your dinner, you have a late night snack, you have like 5 snacks in between. So we essentially never experience ketosis, day in, day out, no ketosis. And for many of us, this has gone on for years and years, or maybe our entire lives. Our bodies have never experienced ketosis. There are lots of new research studies out there that suggest that ketosis is really important in sort of cleaning up our bodies, cleaning up all the waste that accumulates. And one of the things that happens during fasting is, the carbohydrate stores are used up first, within a day or so, they're gone. Then the fat stores kick in and they provide the energy, for days to come. And that just depends on how much fat tissue a person has. And only much later, sort of as an emergency reaction, the body taps into the protein that our body has. And our protein is actually not stored as an energy store. It's just kind of like the emergency cannibalizing that has to be done. If somebody is starving for a month or something, let's say, rather than dying, the body decides, all right, let's waste my muscle a little bit. At least I might survive this. And that's when that kicks in. Muscle wasting does usually not happen in a short-term fasting procedure. All right.
Okay. There's just a couple more. One is to do with a question related to metformin, which we know is in trial at the moment. Still waiting for some results from that. And asking you to comment on how that pathway mimics the keto and the fasting diet, if at all, I guess.
Mm-hmm. Yeah, so how does metformin mimic the fasting diet? So metformin acts on an energy sensor in the cell. It's called AMPK, which then in turn regulates mTOR. And it's all kind of part of this big pathway. That's probably a reason why the metformin animal studies looked very promising, because you're giving a drug that tells the cells, or kind of artificially tells the cells, hey, I'm starving. So that's something that can probably impact essentially on the same pathways that we're looking at when we induce ketosis. It's probably not the entire answer, because metformin acts on this one molecule, whereas actual dietary induced ketosis probably has many more effects that kind of impinge on many different pathways. Yeah.
A question here about creatinine. So in the tested animals, did the creatinine level go down? Did their kidney function improve, therefore? Because there have been lots of studies showing that cyst growth has been reduced, but may not have had an effect on kidney function. So did you measure creatinine?
Yes, we measured creatinine in some of our rat studies, and depending which study it is, it improved or kind of stayed the same. And part of one reason why oftentimes we may not see a very big impact on creatinine in this particular rat model is because there is not much of an impact on kidney function during the age of our experimental window. So the kidney function is only slightly compromised in these rats at that time. So there's only like a little bit of a change in creatinine. So it's a little bit hard to say, how much of a decrease there might translate to some, a human impact there.
Mm-hmm. Mm-hmm. So if you're doing a human trial, you would be looking for some impact on kidney function as well as cystic growth?
Right. Well, so cystic growth in a human trial is essentially means you look at total kidney volume, because you can't take out somebody's kidney and say, hey, let me take your kidney and slice it up and look at it. So this can only be done really by MRI scanning, measuring the total kidney volume. There are some more recent methods on actually measuring cyst volumes out of MRI scans. And that's definitely something we'll be very interested in looking at. The initial clinical trial we're planning is probably going to be too short term to be able to hope to see any effect on cystic volume or kidney volume or even kidney function. Because as I mentioned, PKD is such a slowly progressive disease. In order to see a difference between a treated group and a control group, you really have to follow them for something like 2 years. And that makes a study very, very expensive. So our initial study would be more of a pilot study and feasibility study. And it would probably not answer yet the question about efficacy.
Okay. So just one final one is a couple of people have asked about children and keto diets. So if you could just give some sort of general comments on that.
Great question. Yes. So there is a form of polycystic kidney disease called Adpkd that affects children, essentially from birth on. So it's a very rapidly progressing aggressive version of the disease. that often leads to kidney failure in very young age. And unfortunately, right now, there's essentially nothing that can be done about it, which is extremely unfortunate. That's absolutely on our radar. We're trying right now to test, at least in animal models, to see if very early administration of our dietary changes or supplements might already have a beneficial effect. Just to have a starting point to think about subsequent human studies, and that's something that's going on in the lab right now. Again, I would say stay tuned there. I think it's a very important question to be able to come up with something that might actually help in children with PKD.
Great. Well, I think that's it. We've come to nearly to the end of an hour and a half. Thank you, Thomas, for that absolutely fascinating and incredibly clear presentation of your research. And I particularly found it very, very absorbing. And we're over in Europe, and we're here to help if you're looking for participants in studies and so on. So I'd like to thank everybody for joining us tonight, and we will be, as I said, uploading the webinar to YouTube next couple of days, and I'll be sending out a message to everyone who attended with the link so that you can find it. Fantastic. Thank you very much.
Thanks. Bye-bye, everybody.
Bye.